The next chapter that we are going to discuss is about genetics. So this includes the core part, which can be learned by both SL and HL students. So basically, this is going to establish like a foundation knowledge for both SL and HL students if they haven't done any genetics classes or genetics topic before. So the first subtopic that is needed to be discussed under genetics is the chromosomes, genes, alleles, and mutations. So as we all know, genetics is the study of inheritance and variation of characteristics that these chromosomes are actually controlling. So since we are just in the first part, we just need to make an introduction of what the chromosomes, genes, alleles, and mutations are. So first things first, we have to introduce the structure of the nucleosome, because the nucleosome contains the DNA double helix, which is being supported by the histone proteins. So actually, nucleosome is being studied more in depth in the next chapter, which is chapter 7. Okay, chapter 7, yeah. Nucleus structure. Since students have already studied the DNA structure, okay, we are able to give or actually show the structure of what a chromosome is if we are able to start from the very unit. Okay, so from the very basic unit of the chromosome, which is the nucleosome. So it is made up of a double helix, a double-stranded DNA, which is supported by a protein bead called histone. Okay, so right after giving or discussing the structure of the nucleosome, we can now discuss the characteristics of chromosomes. So, for students to have a more clear idea of what the chromosome should have, we have to start that each and every species, each and every organism have a specific or fixed number of chromosomes. So in the case of humans, we have 46 chromosomes, and they always occur in pairs. So we have 23 pairs of chromosomes coming from each parent. So in a diploid organism, then we have 46 chromosomes all in all. So we have to include the number of chromosomes, okay. It's not really required for students for them to know like how many chromosomes that each and every species should have, but in the case of humans, they need to know that there are 46 chromosomes and then 23 pairs. Okay. Okay, so we also need to include the shape of chromosomes. In the case of the shape of the chromosomes is made up of a very long and thin thread of DNA strands. Okay, that is actually, there is a structure, there is a part within the chromosome strand that is very narrow. We call that as a centromere. Each and every chromosome has a centromere, and they are placed in any part of the chromosome, depending on the length of the chromosome. So we also need to introduce about homologous chromosomes. So when you say homologous chromosomes, they are actually structurally similar. Okay, so structurally similar chromosomes that contains the same set of genes, but they are coming from different parents. So in the later subtopic, meiosis, we should show in a diagram that meiosis is occurring because the homologous chromosomes that are being paired up together coming from both parents, coming from the mother and coming from the father. Okay, so the reason why they line up together is because they occur in homologous chromosomes, or they occur in pairs, having the same set of genes. Okay, so we should also include that chromosomes, it is very important for students to know that chromosomes hold genes. And genes, they are the heritable factors that is being transferred, or that is being inherited by offsprings, which is going to code for a specific characteristic. Okay. So, and each gene are actually located in a specific locus or a specific loci. Okay, loci, it means this is actually a specific area within the length of the chromosome. Okay, so the next, of course, we need to introduce some basic terms that is going to be used fairly in the topic genetics. You have to define what a gene is, the difference of genes, alleles, okay, and genome. Okay, so in the case of genes, genes are actually specific sequences of bases that will code for a specific characteristic or a specific protein. In the case of alleles, this is the alternative form of a gene. Whereas for the genome, if we are going to collect all genes together, the totality or the overall number of overall genes are what we call the genome. Okay, so right after the definition, okay, right after knowing the differences between a gene, allele, and genome, okay, we can now move on to the mutation part. So a mutation is actually a structural change that is occurring in the DNA. It can be in the form of chromosome mutation and gene mutation. So this is one of the common questions that is being asked in IB, wherein they need to differentiate chromosome mutation and gene mutation, and then followed by an example that is being asked for each. So in the case of chromosome mutation, they are the change in the number, okay? Change in the number of chromosomes. Okay, so for example, if humans have 46 chromosomes, then anything that is less or anything that is more than 46 is considered as a chromosome mutation. An example of which is Down syndrome. Okay? So later on, in the part of meiosis, we can discuss why Down syndrome is occurring. So in the case of gene mutation is a mutation that is occurring whenever there is a change in the base sequences. Okay, so for example, if we have, in a normal gene, we have the base sequences of CTC, and then the base T, okay, the base thymine is being replaced by another nitrogenous base. It is going to be replaced by nitrogenous base A. So therefore, this codon is going to code for a different protein, which is supposed to be, in a normal gene, is supposed to be coding for glutamic acid. And then since the nitrogenous base T is going to be replaced by a nitrogenous base A, it will code for a different amino acid, which is valine. So this actually is an example of sickle cell anemia, wherein of course, red blood cells is made up of sequences or groups of amino acids that is going to be condensed together. So if there is a mistake in the assembly of the amino acids, okay, of course, it's going to affect on how the protein is going to fold, or the structure of the protein is going to be affected. Therefore, the function of this specific structure is going to be affected as well. Okay, so change. Okay, so we have to include as well some examples for each. Okay, and if we can provide diagrams as well, then it's going to help the students to better understand on how these types of mutations are occurring.